Hello everyone, I'm Shangua. I'll introduce my work published in iMeta, named A Neural Network-Based Framework to Understand the Type 2 Diabetes-Related Alteration of the Human Gut Microbiome. The gut microbiome has been identified as playing a central role in human health and T2D. Identifying the biomarkers may provide opportunities to get valuable insights into how the microbiota is associated with the related human disease. A common practice is to utilize statistics-based strategies to identify the biomarkers. According to the significance of the difference between the case and control samples. Based on the independent or linear assumption. Deep learning-based methods shown promising performance in the field of biology. Require massive samples for training. Black box, nature, it is difficult to interpret the results. In this study, we proposed a framework combining neural network and random forest for identifying the type 2 diabetes, T2D, related biomarkers and analyzed the identified biomarkers. First, we obtained the species level and gene level profile from the microbial metagenomic sequencing data by calculation. Then, to address the issue of the black box, we employed random forest method for ranking all input features according their importance. Subsequently, we used the NN-based classifier for determining the feature subset with the best prediction performance to be the biomarkers. Next, we constructed the direction interaction network of our selected biomarkers for further analysis. Specifically, among our selected 40 species-level biomarkers, the abundances of the 24 species varied little between case and control samples. However, some of them have been identified to be related to T2D in previous studies. We constructed the interaction network of our selected biomarkers, and found that several biomarkers interacted with many other biomarkers although the abundance of them varied little across all samples, example, coli and S. salivarius, where they are likely the main drivers of the network. The result also suggested that the interaction impact of multiple species rather than individual species may play the important role in the T2D-related alteration. We did the regression analysis on several potential causative factors of T2D, and found that FBG is the most significant factor associated with the human gut microbiome represented by our selected microbial markers. To explore how our marker species covary with the dynamic change of FBG in the development of T2D, we plotted the heat map of mean relative abundances of the marker species at 4 intervals of FBG. The results showed that 24 marker species that do not have significant differences in relative abundance between the case and control samples exhibited diverse patterns. Our analysis suggested that these marker species were likely to play a different role in different stages of T2D development, and then the cumulative effects of these markers derived the T2D-related alteration of the human gut microbiome. Take-home messages. A framework combining neural network and random forest for identifying the type 2 diabetes, T2D, related biomarkers. Constructing the directed interaction networks of the biomarkers for analyzing the potential drivers of the microbial community associated with T2D. Analyzing the covary of the biomarkers with the dynamic change of fasting blood glucose in the development of T2D. iMeta, a journal to change the world.